Our final speaker of the evening is Matt Rendon, and he is going to tell us about his work at Columbia University Medical Center in the Michael Boland Lab. And the title of his presentation is An Organoid Model of Periventricular Nodular Heterotopia.
Who in this room knows someone that has epilepsy? Show of hands. Epilepsy is an incredibly common disease. At least 1 in every 26 people will develop epilepsy at one point in their life. The reasons for this is because epilepsy can be caused by a variety of different things, ranging from genetic reasons all the way to as a result from trauma. Since epilepsy can be caused by so many different things, there isn't one single cure that will cure epilepsy or its symptoms. My research over the summer focused on studying periventricular nodular heterotopia, which is a neurodevelopmental disease associated with epilepsy. Here we see an image of a healthy brain. In the brain, there are these structures called ventricles. Ventricles are where cells originate during neurodevelopment, and from there they migrate outwards to form the properly structured brain. In a brain afflicted with PNH, however, there's defects in the ventricle structures, seen by the clusters of cells around the ventricles. That gives the disease its name. Peri means around, ventricular has to do with the ventricles, nodular means clusters, and heterotopia is cells not where they're supposed to be. So altogether, it's clusters of cells around the ventricles that aren't supposed to be there. The interesting thing about PNH, though, is that it doesn't change brain function. Most neurodevelopmental disease have effects with brain function, but PNH doesn't. The main symptom of PNH is epilepsy, and it is associated with severe epilepsy. Now that we understand that PNH is a disease that affects development, particularly neurodevelopment, we need to understand the processes of neurodevelopment. Again, we have our brain, and cells there originate in the ventricles. From here, they migrate outwards across cell scaffolding to the proper regions that they must reach at the end of their development. For our case in PNH, the cerebrum. Cells are structured in a way that allow them to attach to these scaffolds and move across them to reach the cerebrum at the end of development. However, in brains associated with PNH, there's defects in the cell structure that prevent them from attaching to the neural scaffold and therefore preventing them from migrating outwards towards the cerebrum. Instead, they stay clustered around the ventricles. Neurodevelopment is incredibly hard to study, and we don't know that much about it. The main reason because of this is because it happens in the womb. Since neurodevelopment happens in a fetal brain, we don't have direct access to understanding the different processes or mechanisms that occur in neurodevelopment, keeping much of our knowledge to neurodevelopment locked away from us. But there have been recent breakthroughs in the ways that we study neurodevelopment through the use of organoids. Organoids are three-dimensional cultures derived from human stem cells, and these stem cells can generate any kind of cell, which can be controlled to generate specific organ regions. In the case of us studying PNH, we control these stem cells to turn into cerebral cells or neural cells. To go over how we derived these organoids, we first had a donor provide us with one of their fully grown adult cells. Through chemical treatment, we turned these cells back into stem cells. These stem cells were then chemically treated again to produce neural stem cells, and through the culturing of these neural stem cells, we were able to produce cerebral organoids that let us study the neurodevelopmental processes. And now that we have a mechanism for which we can study neurodevelopment, we can start to look at the genetic causes that might cause PNH. In particular, we look at FLNA. FLNA mutations account for up to 20% of all PNH cases, and is the most common mutation for PNH. It causes PNH because the mutations in this gene deteriorate cell structure, therefore preventing proper formation of neurons, which prevent it from attaching to the scaffold and preventing it from migrating outwards toward the cerebrum. Recently, there have been studies that have discovered a potential gene that might have an association with PNH. This gene is the MAP1B gene. The reason we think MAP1B has an association with PNH is because Mutations in this gene also cause cell structure deformation, and instead of producing properly structured neural stem cells, we instead get cells that are unable to attach to the scaffolding and therefore unable to migrate outwards. Now that we understand what our gene of interest is, we get into what I was looking at over the summer in my research. We wanted to define the role of MAP1B in PNH. We first wanted to see if developmental defects appear at all, and if we see these in the organoids. Then, if these developmental defects are there, we wanted to see at what level they appear. In order to study these different questions for MAP1B, we had to generate specific organoid lines that showed mutations versus healthy organoids. To recap how we get organoids, we have an adult donor provide us with one of their cells, which we then turn into a stem cell. From here, if we want to show proper development, we can turn these stem cells into healthy neural stem cells. These neural stem cells then produce control organoids, or organoids that don't have any mutations. But at the stem cell level, we have another path we can take. We can induce mutations in the stem cells to produce mutated neural stem cells, which we can then turn into either FLNA organoids, which we know is a disease associated with PNH, or the MAP1B organoids, which is our gene of interest that we're looking to study. With these three organoids, we were able to grow them and first view the whole organoid development to try and see if there were any defects at that level. We took bright field images every 2 days, and specific time points are shown here that will show defects in these organoids. At day 4, we start to see the cells divide and turn into the organoid structure. On the far left, we have our control organoid, which is our healthy development and the unmutated one, followed by the FLNA, which is our known mutation, and the MAP1B samples, which is our gene of interest. At day 10, we start to see a disease phenotype. We start to see improper formation of organoids. We start to see morphological differences, and that is seen through the protrusions and the bumps on the organoids that are the mutated ones, as opposed to the control cells. At day 18, we start to see a different phenotype. We start to see transparency in the organoids, which is indicative of decreased levels of cell division. Since we see these defects in both the FLNA and the MAP1B samples, we understand that MAP1B is correlated with FLNA and therefore can be correlated with PNH causation. Next, we wanted to see the internal mechanisms and the internal developmental processes that might be affected by PNH. For this, we took these organoids, preserved them, sliced them, and were then staining them to see the internal structures. We used the fluorescent imaging to stain for 3 particular things. First, the ventricle structures, which again are the regions of the brain where cells originate and migrate outwards from. We then stained the neurons to show more clearly the ventricle structures as well as the migratory path of these cells through development. And finally, cell nuclei as a counterstain in order to see the red and green staining more clearly. In our normal sample, we are able to see proper formation of ventricles. These are these red ring structures surrounded by neurons running parallel to them. These ventricle structures indicate proper development and will allow for proper migration of the cells during development. Our FLNA sample, however, shows a clear defect in the ventricle formation. We see that there is both a decrease in size as well as a decrease in number of ventricle structures in the disease sample, which we know would impair cell migration and therefore cause PNH disease symptoms. The MAP1B sample shows a similar phenotype to the FLNA, further confirming its role as a PNH causing mutation. We see a decrease in the size of these ventricles as well as a decrease in the number of the ventricles throughout the entire organoid. Finally, we wanted to look if at the individual neuron level there were defects. For this, we took the organoids and dissociated or broke them down to see the individual cells through bright field imaging again. These dissociated neuron cultures, so first we get to see the healthy neuron culture, which we see through the neurons that have rounded cell bodies, as well as extensions that are unfragmented and proper, like proper formation of the unfragmented extensions. The FLNA sample, however, has these neurons, but instead there's fragments in the extensions. This would prevent proper cell signaling, which would then in turn prevent proper signaling of the brain and cellular processes. The MAP1B samples also show fragmented neurons, which is again a symptom corresponding to the PNH disease. We also see a very unique mutation in the MAP1B samples. We see the emergence of an abnormal cell type. In the conditions that we set for these cultures, no other cell other than neurons should be able to survive or divide. However, the MAP1B samples produced an abnormal cell type that shouldn't exist in the first place. We don't understand the direct mechanism that caused this abnormal cell type growth, and we don't understand what the abnormal cell type is, but there's potential that it could be associated with PNH. But due to the fact that we see unfragmented neurons in the MAP1B sample, that we can correlate it with FLNA and therefore correlate it with PNH. Our 3 experiments were able to show us that MAP1B mutants show defects in cortical development associated with PNH. Our study of the whole organoid and bright field imaging showed decreased cell division rates as well as abnormal cell growth and abnormal organoid formation. Looking at the internal structure, we were able to see through fluorescent staining that there's impaired cell structure in development, which was correlated with the loss of ventricle structures and the decrease in size. Then, at the individual cell level, we were able to see that neurons have unhealthy structures, as well as, in the MAP1B specifically, the emergence of an abnormal cell type, which we aren't sure whether or not it has a causation in PNH. We're hoping that this research moving forward, that we can do more additional experiments to further verify the MAP1B phenotypes. We want to be able to further confirm if MAP1B causes PNH, as well as look into what exactly is the abnormal cell type and the effect that it may have on this mutation. Next, we want to use this research as a means to optimize the use of organoids in disease research. Organoids have only been used in the past 5 years, and therefore, not all of the mechanisms that define organoids are fully understood. And finally, we hope that our results can ultimately translate to either drug screening or gene editing in an attempt to solve PNH and the symptoms it causes. We hope that our research will be able to take these disease organoids, and through the application of different drugs or direct genetic editing, we can in turn cause healthy organoids and therefore cure PNH. I'd like to thank a few people that I worked with over the summer at Columbia University. My mentor, Dr. Sarah Song, my PI, Dr. Michael Bolin, for letting me work in the lab over the summer, and Bright YFA for working closely with me for 10 weeks. Also, I'd like to thank Dr. Selena Gell, my SRP bio mentor, for always pushing me to do my work. My SRP bio family for struggling with me for the past year and a half, and my friends and family for the constant support. I'll now answer any questions.
So, when you were developing the organoids, what was the reasoning behind chemically treating mature cells from the samples instead of just directly taking stem cells that were already there? Was there specific instructions?
So, since PNH is a ultra rare disease, is the clinical term for it, there isn't really that many people that are either well enough, or we don't have contact with people that will be able to give us their direct stem cells, which would then have their genetic mutations in the organoid cultures. So instead, we decided to take regular adult cells and therefore treat them to turn them into stem cells and generate the organoids that way. So we could keep kind of a baseline through our mutation, through our different samples, and be able to see if the mutation specifically has these phenotypes, rather than it being a cause of genetic variation.
How did you introduce the mutation in the neural stem cells?
So we actually introduced the mutation in the stem cell level. So we used genetic editing to change the DNA to produce the MAP1B or the FLNA mutation, and then from there we treated them chemically to produce the neural cells, and then from there it would either be a healthy neural cell or a mutated neural cell.
Why specifically epilepsy? I feel like the pictures that you showed us seemed like a pretty major issue, and like could this potentially be the cause of something else, like other diseases other than epilepsy, or is epilepsy just the main focus of the study right now?
Yeah, so PNH itself doesn't really have other symptoms. I mean, interestingly, it doesn't have any defects in like brain function, and the main symptom of it is epilepsy. But the defects in the neurodevelopmental processes is definitely seen in a wide variety of different diseases. And we wanted to first understand the mechanisms in PNH, and then from there, try and see if we can broaden it to other neurodevelopmental diseases.
Do you or your team have any hypotheses as to what this abnormal cell that you've observed is perhaps similar to or functions as?
We're not entirely sure. So since the organoid studies are really new age, and especially like the MAP1B, since it's still being defined as a causative mutation, we're not entirely sure the effect that it has. Organoids aren't the best at showing like signaling in the brain, since they are not like connected to a vascular system or any type of functionality like that. So we're not able to see like the functionality of these abnormal cells, but we'd have to look into that further.
So I believe, the goal of this is to hopefully develop a cure for PNH. Why could that not already be done from just the FLNA mutation? Why do you also need this other MAP1B mutation?
So since the FLNA mutation only accounts for about 20% of all PNH cases, there's different mutations even within the FLNA, like different amino acid changes that could cause PNH. So there's a lot of different individual changes that could cause PNH that we can't cure all of them, since they all require a different treatment developing for them, and therefore there's that 80% that aren't identified mutations that cause PNH. So we can't really develop a cure for PNH broadly.
So what causes the gene mutation of MAP1B and the other genes?
So genetic mutation can either happen naturally or it can be inherited. So a lot of the cases that we do see is parents that have the genetic mutation that go undiagnosed and pass it to the kid, and through just the mixing of genetic components with the other parent, that it might show severe symptoms in the kid. So they can either be induced naturally through just the way that cells divide and just the genetic mutation that happens there, or it can be inherited through the parents.
How does the rate that the organoids develop as you observe them compared to how it would normally develop inside of the fetus?
So the organoids aren't able to show one specific cerebrum. So instead, the ventricle structure would only be, as we saw in the pictures of the healthy brain, like those two regions in the brain, but rather we see a bunch of mini circles and or ventricle regions in the organoid, which kind of just shows us the broad term developmental processes, but it doesn't specifically show us like one ventricle equals one cerebrum. So we aren't able to determine the growth rates and the different cell division rates as compared to like in fetus.
All right, well, let's thank Matt.